Okay, so welcome back to this next video in which we are discussing calcium signaling. Okay, right. So we're currently in the process of discussing IP3 receptor mediated calcium signaling. Okay, so we've now discussed the generation of IP3. We've discussed how phospholipase C enzymes, activated by different mechanisms, can then hydrolyze phosphatidylinositol 4,5-bisphosphate molecules to produce diacylglycerol and IP3. The diacylglycerol will remain in the plasma membrane and will come back to its signaling role later on. And the IP3 can drop into the cytoplasm, and we now want to see what the IP3 is actually going to do to activate calcium signals. Okay, and it is going to work by having an effect on the IP3 receptors. Okay, so the next topic it's going to be important for us to take on is the IP3 receptors. Okay, so we want to study the structure of these things, and we then want to study how these things can be activated to open, basically. Okay, so firstly, what actually are the IP3 receptors? So the IP3 receptors are calcium channels that are present in the membrane of the endoplasmic reticulum. Okay, and when they open, they are going to allow calcium in the lumen of the endoplasmic reticulum to move out of the lumen of the endoplasmic reticulum and into the cytoplasm of the cell, okay, and therefore produce an intracellular calcium signal. Okay, right. So let's start off then by discussing the structure of these IP3 receptor channels. Okay, right, so I'll start off by drawing the ER membrane here as a phospholipid bilayer. Okay, and then sitting in the ER membrane here, we're going to have our IP3 receptor here. Okay, and it is a calcium channel, so here is the pore down the middle through which calcium ions can move from the luminal side to the cytoplasmic side. Okay, so we'll say this is the luminal side, and this is the cytoplasmic side of this membrane. Okay, so let's just put a little bit of colour onto the picture. Okay, like so. And the abbreviation that people often use for an IP3 receptor is IP3 and then capital R, like so. Okay, so this is our IP3 receptor here. Okay, so let's now talk about the structure then of these IP3 receptors. Okay, so the first thing to say is that IP3 receptors are absolutely not just one protein. Okay, instead they are made up of multiple proteins stuck together. Okay, so the IP3 receptor is actually made up of 4 separate subunits joined together. Okay, that each make up a quarter of the entire thing. So basically, the IP3 receptor is a tetramer. Okay, and this isn't controversial. Okay, so we saw that the crack channel being a tetramer was controversial, but the IP3 receptor being a tetramer is broadly accepted. Okay, right. Now, there is a complexity to the IP3 receptor that the crack channel didn't have, okay, which is that the IP3 receptor subunits, which are these proteins which make up a quarter of the IP3 receptor, there is not just one of them, basically. Okay, in the case of the crack channel, there was only one protein that you could use as the subunit for a crack channel, which was that aura-I1 protein. In the case of the IP3 receptor, there are 3 different types of subunit that you can use to build an IP3 receptor. Okay, so the really simple thing would be if there was just one subunit for IP3 receptors, which was called the IP3 receptor subunit, and all you had to do was make 4 copies of that, stick them together, and then you've got your IP3 receptor. However, it's not the case, basically. Instead, there are 3 different types of IP3 receptor subunits, and let me tell you what their names are. So there is the type 1 IP3 receptor subunit, which is called the IP3R1 protein. So this is the type 1 subunit of an IP3 receptor. Okay, then there is the type 2 IP3 receptor subunit, which is denoted IP3R2. And then there is the type 3 IP3 receptor subunit, which is denoted IP3R3, like so. So these are the names for the 3 different types of subunit for IP3 receptors, basically. okay, and they are all coded for by different genes. So this means it's more complex with regards to how to actually assemble an IP3 receptor tetramer, because now we have these 3 different IP3 receptor subunits to choose from, and we have to fill 4 sockets with these IP3 receptor subunits. Okay, so how do you do it? Is it the case that you can only build IP3 receptor tetramers where the 4 subunits are identical? Okay, well those sort of tetramers are called homotetramers. Okay, homo means the same. Okay, so a homotetramer is where all 4 subunits are identical. And indeed, each one of these types of IP3 receptor subunit can form homotetramers. So you can build an IP3R1 homotetramer where you have 4 IP3R1 subunits in each of these 4 sockets. Okay, you can build an IP3R2 homotetramer where you have 4 IP3R2 subunits in these 4 sockets, and you can build an IP3R3 homotetramer where you have 4 IP3R3 subunits in those 4 sockets. Okay, but you can also do the more complicated thing, which is you can build IP3 receptor tetramers which do not have all 4 subunits the same. Okay, and these are called heterotetramers. Okay, hetero means different. Okay, so a heterotetramer is one where the subunits of the tetramer are not all the same, they're different basically. Okay, now, I would love to be able to tell you which heterotetramers you can build, okay, and exactly what the arrangement of the different subunits in the heterotetramer are. However, that sort of information is just not known. We know that heterotetramers can form, okay, but we don't know which ones can form at the moment, and we don't know what the arrangement of the subunits in these heterotetramers actually is yet. That's an area of ongoing research basically, and in the future we will have that sort of information available. But at present, I can tell you that it's known that you can form IP3 receptors which are not homotetrameric, but I can't tell you exactly what the stoichiometry of the different subunits in them is, and what the arrangement of the different subunits in them is. Okay, right, so then that's what we need to know about the structure of an IP3 receptor then. It is this channel in the ER membrane which can open and allow calcium out of the ER lumen into the cytoplasm. Okay, now it's not normally open. Okay, so I now want to discuss how can you induce this thing to open. Okay, and this is subtle, this is clever, okay, and I should say right now that we don't fundamentally know for certain that what I'm about to tell you is true. What I'm going to tell you is a very good model for how IP3 receptors are opened, okay, and we can use this model to explain the 2 different types of calcium signal, which are calcium puffs and calcium waves. However, at present we do not have the full structure of the IP3 receptor published yet. Okay, so we don't have an X-ray crystal structure, okay, and we don't have other types of structure that have been published yet, okay, so we certainly don't know the gating mechanism of IP3 receptors on the level that we know, for instance, the gating mechanism of voltage-gated ion channels, or of cis-loop ligand-gated ion channels, or anything like that, where you can actually follow which bits move, basically. We don't know that yet. Okay, but we do have models for how these things open, okay, which explain the different types of calcium signal that can be produced. Okay, and it's not as simple as you might think. You might think that IP3 just binds and that causes it to open, but it's not the case. IP3 does not directly cause the channel to open. Okay, so let me now discuss then this model. Okay, so for this purpose, what I'm going to do is I'm going to draw the IP3 receptor tetramer as viewed from above. Okay, so we're now looking down from above, or from the cytoplasmic aspect, okay, and we're looking down at the top of our IP3 receptor tetramer here. Okay, so here are the 4 separate subunits of the IP3 receptor tetramer then. Okay, now each one of these IP3 receptor subunits is going to have a binding site for IP3. Okay, so I'll represent this by this orange dot here. Okay, and I want to stress that this is a picture. Okay, this is a little picture to illustrate a point. Okay, it's not supposed to be taken literally that that is the position of the IP3 binding site, so it's just a picture. It's not meant to actually resemble how the IP3 receptor actually looks. As I say, we don't have the structures yet of the IP3 receptor. Okay, right, so these are the binding sites for IP3. Okay, so the IP3 binding sites are represented there in orange, and every single one of the 4 subunits of the IP3 receptor tetramer will have its own IP3 binding site, basically. Okay, right. However, each of these IP3 receptor subunits also has 2 other important binding sites that I need to tell you about before we talk about what the IP3 binding site actually does when IP3 binds to it. Okay, and these other 2 binding sites are for calcium. Okay, so I'll represent one of these calcium binding sites in blue here, and this is going to be an inhibitory binding site. Okay, so this is what's called the calcium inhibitory binding site. So calcium can bind to this inhibitory binding site. Okay, and when it does so, that absolutely makes sure that the channel does not open. Okay, so it keeps the channel closed if calcium is bound to this inhibitory binding site for calcium. Okay, so this is our calcium inhibitory binding site. Okay, represented there in blue. Okay, we also have a stimulatory binding site for calcium. Okay, which I'll represent here in red. And if calcium binds to the stimulatory calcium binding site, it will cause the channel to open, providing that you don't have calcium bound to the inhibitory binding site. If you have calcium bound to the inhibitory binding sites, then even if you have calcium in the stimulatory binding sites, it won't open, basically. The inhibitory binding site is stronger than the stimulatory binding site. So the ideal thing that you need is calcium just bound in the stimulatory binding sites and not in the inhibitory binding sites. Okay, so this is the calcium stimulatory binding site. Okay, so we'll colour that one in red. Okay, right. So this is the idea then. Let's say that we have not got any IP3 being produced yet, okay? So there's no IP3 in the cytoplasm, okay? There's not any IP3 bound to these 4 IP3 binding sites, okay? In this pre-IP3 state, the inhibitory calcium binding site here, these binding sites in blue on each of the 4 IP3 receptor subunits, these have a high affinity for calcium, and the stimulatory calcium binding sites have a low affinity. Okay, so initially when there's no IP3, these ones have the high affinity and these ones have the low affinity for calcium. Okay, so I'll colour these in yellow, and these are the pre-IP3 states, basically. Okay, so pre-IP3 you have high affinity state for the inhibitory calcium binding site and a low affinity state for the stimulatory calcium binding site. Okay, right, so what that means is that the calcium in the cytoplasm, and remember you do have some calcium in the cytoplasm, it's not 0 the concentration of calcium in the cytoplasm, okay, it is 100 nanomolar, okay, so what's now going to happen is you're going to end up getting calcium ions bound to the inhibitory binding site, basically, because that's the high affinity one, but you're not going to get any calcium bound to the stimulatory ones because that's got a low affinity. Too low for calcium to bind there at this low 100 nanomolar concentration of calcium that we have normally in the cytoplasm of the cell. Okay, so that means that normally the calcium in the inhibitory binding sites is keeping the IP3 receptor continuously closed, basically. It's stopping it from adopting the open conformation. So the channel will have its gate shut, whatever that gate may be, and we don't know what the reality of the gate actually is at the moment. But something is blocking calcium from actually moving through that pore. Okay, right, so that's what happens pre-IP3. Now let's say we have produced IP3. So IP3 has now arrived. Okay, so now we're in the IP3 era. What happens is this reverses. Okay, so the calcium inhibitory sites, well, whoops, I haven't actually said that IP3 binds yet. We'll come to that. Okay, so when IP3 goes up in the cytoplasm of the cell, what's now going to happen is you're going to get IP3 binding to these IP3 binding sites here, okay? And it's not clear as to whether you need IP3 to bind to all 4 of the IP3 binding sites of the IP3 receptor. However, to keep this simple, let's imagine that all the 4 of the IP3 binding sites need to get IP3 bound to them. Okay, so let's say 4 IP3 molecules are coming in, 1 will bind to each of the 4 IP3 receptor subunits. Okay? What that is now going to trigger in each of these IP3 receptor subunits is a conformational change that results in this calcium inhibitory binding site losing its affinity for calcium. Okay, so now once you've got IP3 bound in these 4 sites, the calcium inhibitory sites on each of the 4 subunits now gets a much lower affinity for calcium. So the conformation of that site changes so that its affinity for calcium hugely decreases. Okay. Meanwhile, the exact opposite thing happens for the calcium stimulatory binding site. Okay, you get a conformational change in the stimulatory calcium binding sites on each of the 4 IP3 receptor subunits, and this now causes them to adopt a much higher affinity conformation, basically. Okay, so it swaps over. The calcium inhibitory binding site now will not bind calcium under the normal cytosolic calcium concentration of 100 nanomolar, but the calcium stimulatory binding site will now bind calcium under that normal 100 nanomolar calcium concentration. Okay, so now calcium ions can bind to the stimulatory binding sites, and you won't get any calcium bound to the inhibitory binding sites, and then the IP3 receptor will open, basically. Okay, and it will then release calcium from the ER into the cytoplasm, producing us a calcium signal. Okay, however, be warned that the activation of the IP3 receptor by calcium is what is known as biphasic. Okay, so this is the genius of this. If the IP3 receptor is now releasing calcium from the ER into the cytoplasm, then the calcium concentration around this IP3 receptor is going to go up hugely, basically. Okay, now with the much higher concentration of calcium, you're not just going to get calcium binding to the calcium stimulatory binding sites anymore. You're going to start getting calcium binding to these lower affinity calcium inhibitory binding sites. Okay, so calciums will now be in both of the sites, and we know what that then causes. It causes the channel to close again. Okay, so this is a nice negative feedback loop where once you've raised the cytoplasmic calcium concentration enough around you, then it's going to actually cause the channel to close again. Okay, and the channel won't be openable again until you've removed those calcium ions from the inhibitory calcium binding sites. Okay, so we can draw then a picture like so. Okay, so if this is the open probability of the channel, so how likely the channel is to be open, basically. Okay, and we're assuming we're now referring to an IP3 receptor with 4 IP3 molecules bound in these 4 IP3 binding sites. Okay, and we then plot calcium concentration on the x-axis down here. We'll get a graph that looks like this, basically. Okay, so at low concentrations of calcium, you can see that the chance of the receptor being open is going to go up, basically. The more calcium you have around the IP3 receptor, the more chance it is to open, because the more is the chance that you'll get calcium binding in the stimulatory binding sites. But raise it too high, basically, and you'll start straying back down in your open probability, because now calcium will start binding to the lower affinity calcium inhibitory binding sites, and that overrides the calcium that's bound to the stimulatory binding sites and causes the channel to close. Okay, so this sort of peak is at around 200 to 300 nanomolar calcium in the cytoplasm. Okay, so if you really want to activate IP3 receptors fantastically, that's the sort of concentration of calcium that it's best to have in the cytoplasm of the cell. Okay, that's the sort of concentration where you can get these stimulatory calcium binding sites all occupied with calcium. So you can get a calcium bound there, a calcium bound there, a calcium bound there, and a calcium bound there, without having the inhibitory calcium binding sites activated by calcium. So 100 nanomolar, the more normal calcium concentration inside the cytoplasm of the cell, will be down here basically. So it's quite difficult actually to activate IP3 bound IP3 receptors with the normal calcium concentration in the cytoplasm, simply because if you've only got 100 nanomolar calcium concentration, and I've put n little m, it should be big N, okay, it looks like nanometers, okay, nanomolar. Okay, so simply because if you've got this small calcium concentration in the cytoplasm, the normal resting concentration, it's quite difficult basically to get 4 calciums binding into these 4 stimulatory calcium binding sites. Okay, and therefore it's quite difficult to get your IP3 receptor to actually open basically. Okay, so to go over this again, what you need to do is you need IP3 to bind to the IP3 receptor binding sites the IP3 binding sites of the IP3 receptor subunits. Okay, they trigger these conformational changes in the IP3 receptor subunits now, okay, which make the inhibitory calcium binding site lower affinity and the stimulatory calcium binding site higher affinity. Okay. Then what you need to happen is you need 4 calciums to come along and 1 to bind to each of these stimulatory calcium binding sites. Okay, and I'll stress again that we don't really know whether it's fully necessary for 4 IP3s and then 4 calciums to actually bind. Okay, but to simplify this down, we'll say that that's the case. Okay. Then what will happen is, if you somehow achieve that, if you do achieve 4 calciums binding in these calcium stimulatory binding sites, and I'll stress that that is not trivial to do, okay, not under normal cytosolic calcium concentration. At a calcium concentration of 100 nanomolar, it's not easy to get 4 calciums binding there. Even once you've got the 4 IP3 molecules, you have to now wait quite a long time basically to get 4 calciums binding there. Okay, and then once those calciums are bound there, then it will finally open. Okay, it will start releasing calcium from the ER lumen. Calcium will build up in its vicinity, and when calcium level gets too high around it, then you'll start getting calcium binding to the inhibitory calcium binding sites, and that will cause the IP3 receptor to now shut again. Okay, and then if you want to reopen it, what you need to do is lower calcium concentration back down so that the calcium comes off these inhibitory binding sites. Okay, now what I want to talk about then is how this gives rise to the possibility for propagation of IP3 receptor opening basically. So let me show you this. So if we have the ER membrane here, okay, and we have 3 IP3 receptors in the ER membrane, okay, and I'll just show 3 here, okay, that are neighbours to one another. Okay, you could have far more than this, just for simplicity I'm showing 3 here, okay, and let's just colour them in to make the picture look more interesting. So here are the IP3 receptors now coloured in orange here. Okay, right, so let's say that each one of these IP3 receptors has now got 4 molecules of IP3 bound to it. So you've had one of these cascades that has resulted in the production of IP3 occur, okay, and each one of them has got 4 IP3 molecules bound to it. So it is now primed and ready to be activated to open by calcium binding. Okay, now we've got normal cytoplasmic calcium concentration of 100 nanomolar. Okay, so that means it's quite difficult for any one of these to get 4 calcium ions binding to their 4 calcium stimulatory binding sites. Okay, so you're going to sit around for a bit waiting for one of them to finally get 4 calciums binding to the calcium stimulatory binding sites. But once one of them happens to do that, then it becomes interesting. Okay, so let's say this one in the middle happens to just manage to get 4 calciums binding to its 4 calcium stimulatory binding sites at the normal resting calcium concentration of 100 nanomolar. It's then going to open, and what does it then do? It opens and allows calcium to leave from the endoplasmic reticulum lumen into the cytoplasm. Okay, and this results in calcium going up here. So now what's going to happen? Calcium is going to spill over onto its neighbours, okay, and now the chance that these ones are now going to get calcium binding to their stimulatory binding sites is going to be hugely increased, because we have increased the local calcium concentration from maybe 100 nanomolar up to maybe this level, 200 to 300 nanomolar. Okay, so now they are going to get calcium binding to their 4 stimulatory binding sites basically, and they're going to be activated to open. So they will then release calcium from the endoplasmic reticulum, and they then might have neighbours next to them. So if I just show this here, here's another one further along, and here's another one further along this way, okay, like so. And what can then happen is they can spill calcium over onto their neighbours, and their neighbours can then get calcium binding to their 4 calcium stimulatory binding sites, providing, of course, that all of these have 4 IP3 binding to them. Okay, so if you get to one that doesn't have 4 IP3s bound to it, then of course it won't have undergone this conformational change where the calcium stimulatory binding site now has the high affinity and the calcium inhibitory binding site now has the low affinity. Instead, it will be in the pre-IP3 state where the inhibitory binding site has the high affinity and the stimulatory binding site has the low affinity. And therefore, when calcium spills over onto them, you're just going to reinforce the block on opening, basically. You're going to put more calcium into their inhibitory binding sites rather than their stimulatory binding sites. But if these do have 4 IP3s bound to them, then of course you'll get 4 calciums binding to the stimulatory binding sites, and therefore the things will open. Okay, so this gives rise then to propagation in the release of calcium from IP3 receptors. The release can propagate basically in this way, and this is going to be the foundations of what calcium waves are all about. Okay, so what I now want to then discuss is the different types of calcium signals, okay, that can result from stimulating a calcium mobilizing G protein coupled receptor, for instance, so a GQ/11 coupled G protein coupled receptor. Okay, so there are these 2 types of very different calcium signals that you can produce, okay, called calcium puffs and calcium waves. So I'm going to start off by telling you about what you have to do to get calcium puffs occurring within a cell. Okay, right. So let's imagine that we are stimulating the cell with some agonist for a GQ/11 coupled G protein coupled receptor. So let's imagine that we're going to stimulate the cell with acetylcholine maybe, and we can be stimulating the M3 receptor, one of the prototypical examples of a GQ/11 coupled G protein coupled receptor. Okay, right. So what we're initially going to do if we want to get calcium puffs occurring in the cell is we're going to stimulate with a low concentration of acetylcholine. Okay, so if I just draw my picture of the cell here, okay, so let's say this is our cell here, okay, and once again I'll draw the nucleus. So here is the inner nuclear membrane, and then we'll have the outer nuclear membrane, and now I'm going to draw the endoplasmic reticulum, and I'm going to make the endoplasmic reticulum bigger now. Okay, so we've got lots of sites for signaling to occur, basically. Okay, so here is my cell, and we've now exposed this cell to a low concentration of acetylcholine, and that's absolutely key here to get these calcium puffs. Okay, so low acetylcholine concentration. So when you put square brackets like that, that means concentration. Okay, so we want a low acetylcholine concentration. Now, in a low acetylcholine concentration, you're only going to get some of the M3 receptors on the surface of our cell here actually getting acetylcholine binding to them rather. Okay, so the cell will have lots of M3 receptors, so I'll just draw these here, and I'll represent them by little purple portions. So let's put the M3 receptors all over the cell here, like so. Okay, so you've got loads of M3 receptors all over the place. But if you have a low acetylcholine concentration, very few of these are going to actually get acetylcholine binding to them. So let me represent the acetylcholine by a turquoise dot. Okay, so let's say this one gets acetylcholine binding to it, and this one gets acetylcholine binding to it. Okay, so at this low concentration, only a tiny little amount of them get acetylcholine actually binding to them. Okay, then what happens is in the little vicinities of these M3 receptors here, you're going to start up this cascade basically. You're going to be activating GQ/11 heterotrimeric G proteins, you'll be activating phospholipase C beta enzymes, which will be breaking down PI45P2 molecules into diacylglycerol and IP3. So you'll be generating IP3 locally, and that's the absolutely key thing about this. We get local signals rather than global signals. So we get IP3 going up locally. Okay, so you'll get IP3 being produced in this little area here. You'll get IP3 being produced in this little area here. Okay, now that's going to lead to local calcium signals. Okay, so what's now going to happen is the IP3 is going to then bind to IP3 receptors in the local regions of the endoplasmic reticulum, so these portions here. So in these portions of the endoplasmic reticulum, shown here in orange, the IP3 receptors are going to get IP3 bound to them, okay, because that's where the IP3 has actually been produced. Whereas in all the other portions of the endoplasmic reticulum, these receptors are not going to get IP3 bound to them. Okay, that then means that in this portion of membrane, all we now need to wait for is one of the IP3 receptors in that orange portion there to get 4 calciums bound to its 4 calcium stimulatory binding sites, okay, under the normal physiological concentration of calcium of 100 nanomolar. And then what will happen is you'll get this propagation of calcium release by the IP3 receptors in that region. So in that region you might have a huge number of IP3 receptors which have all got 4 IP3 molecules bound to them, and then all you're waiting for is just one of them to happen to get 4 calciums bound to it at the normal 100 nanomolar calcium concentration in the cytoplasm, and then the thing will spread in the way that I've shown you here. It'll propagate to its neighbours, and on it will spread. Okay, right. However, it will not spread beyond this region because it will hit the point here where you get onto IP3 receptors which do not have 4 IP3 bound to them. Okay, so let me just draw this here. So this is now an IP3 receptor outside of that orange zone, which I will now colour in a different colour. So let's have it represented in green here. So this one has not got 4 IP3s bound to it because it's not been exposed to enough IP3 basically. And now when calcium spills over into this IP3 receptor space, okay, it will not cause this IP3 receptor to open, okay, simply because, remember, when you don't have the 4 IP3s bound, it's the inhibitory binding sites which have the high affinity, and the stimulatory binding sites have the low affinity. So now you're just going to get calcium binding to the inhibitory binding sites. So basically the IP3 receptors in this little orange region of the ER membrane are going to all open and release calcium, okay, into this local region, but you're not going to get IP3 receptors in the neighbouring portions opening, and therefore you're not going to get calcium going up in these neighbouring portions. Okay, so now let me show the calcium. Calcium can be represented as this red dot here. You're going to get calcium appearing in that little area there, and also in this little area here. Okay, so these are local calcium puffs, as they're called. Okay, so this is what is meant by a calcium puff, this local rise in calcium, and it's transient. Okay, so let's put some other key properties here. It's a local rise in calcium, and the last key word is that it's transient, it's short-lived. So it's a little puff, it's going to be a flash of calcium basically. Calcium will go up and then it will go down. So why is it going to be transient? Okay, well, when an acetylcholine binds to the M3 receptor, it doesn't stay there forever basically. Okay, it will cleave off eventually. So let's say this acetylcholine now has cleaved off. Okay, that will mean that the M3 receptors will go back to being in the inactive conformation, so they'll stop activating more GQ/11 heterotrimeric G proteins. Okay, so we're no longer getting any more GQ/11 heterotrimeric G proteins activated here. Okay, the ones that we have already activated will turn themselves off, and this is something that I haven't actually mentioned before. Okay, so I'm bringing back up my pictures of heterotrimeric G proteins, specifically the alpha subunits. Okay, and we'll have a look at how they can turn themselves off. So, the alpha subunits of heterotrimeric G proteins, and we'll do it on this picture here, they are actually enzymes. Okay, and what sort of an enzyme are they? They're a GTPase, basically. They are capable of catalyzing the hydrolysis of GTP down into GDP and inorganic phosphate. Okay, so they are capable of turning this GTP that they have bound here, which is keeping them in the on state, into GDP and then inorganic phosphate. So when they do this, they will keep the GDP bound to them. Okay, and then they'll go back to being in the off state, and they'll spit the phosphate group out into the cytoplasm. Okay, so basically all the alpha subunits, including the alpha Q/11 subunits, are GTPases. They have this intrinsic GTPase activity, and they're therefore capable of converting this GTP that they have bound to them in the on state back into GDP and releasing the phosphate into the cytoplasm. Okay, when the alpha subunit has GDP bound to it, then it will cleave away from the phospholipase C beta enzyme, okay, or whatever other target it might have. So if it's an alpha Q/11, the target will be a phospholipase C beta enzyme, but obviously for other alpha subunits it will be a different target. But once it's back in the off state and has GDP bound to it, it can no longer bind to its target. So it will cleave away from its target, and it will now want to rebind to the beta-gamma subunit. So it will go and find a beta-gamma complex, basically, and it will rebind to it, and therefore you'll turn the heterotrimeric G protein back into this off-state heterotrimeric G protein, where the alpha subunit has GDP bound, and it's bound, therefore, to the beta-gamma complex. So basically, the message there is that eventually the alpha Q/11 subunits turn themselves off, basically, and stop activating phospholipase C beta enzymes, and therefore stop producing more IP3. Okay, right. So once you have stopped activating more alpha Q/11 subunits by the M3 receptor no longer having acetylcholine bound to it, then the alpha Q/11 subunits will hydrolyze their GTP eventually, and they'll turn themselves off. Therefore, you're going to no longer have any phospholipase C beta being active. You're not going to be producing any more IP3. Okay, so no more IP3 is being produced. The IP3 that has been produced will be degraded very rapidly. Okay, so now the IP3 receptors are going to lose the IP3 that's bound to them. In addition, what you have to remember is that when the calcium rises up in this level to a higher level here, what will happen is it will start inhibiting the IP3 receptors, whether they have IP3 bound to them or not, basically. Okay, because when calcium gets too high in the vicinity of the IP3 receptors, then it doesn't matter that the IP3 has lowered the affinity of the inhibitory calcium binding site. If it's too high a concentration of calcium nearby the IP3 receptor, calcium is going to bind to that inhibitory calcium binding site anyway. Okay, and therefore even if the IP3 receptor does have IP3 bound, it's going to be turned off, basically. It's going to be closed because of the high calcium concentration nearby. Okay, so that then is how you can stop the release of calcium. Okay, and then what of course will happen is the calcium that has been released into the cytoplasm will be returned into the endoplasmic reticulum and pumped out of the cell by the mechanisms that we've discussed earlier. So SERCA will return it back into the endoplasmic reticulum, the PMCA, the NCX and the NCKX will chuck it out of the cell and therefore reverse the calcium signal. So this calcium signal will appear and then it will vanish basically, and that's the concept of a calcium puff. Okay, so it's a very local calcium signal. Now if you maintain this cell then in the acetylcholine at this low concentration, what will happen is you'll continually get different M3 receptors having the acetylcholine bound to them. Okay, so the acetylcholine might cleave off this one, but now this one over here might get a acetylcholine molecule binding to it, and that will produce a calcium puff here. So if you watch the cell and watch its calcium signals, you'll see calcium puffs appearing all over the place at different times basically. So it's just calcium puff after calcium puff after calcium puff in different little local locations within the cell basically. So these calcium puffs are very transient calcium signals that occur very locally. Okay, in the next video what I want to turn my attention onto is the other type of calcium signal, which is a calcium wave, which is a global calcium signal rather than a local calcium signal.